Hello everyone, welcome to Citizen Pro channel. In today's video, we are going to dive into pharmacovigilance. Pharmacovigilance is a field that safeguards the safety of medicines to all of us. Whether you are a patient, parent or a medical healthcare professional, was simply curious about how drug safety is ensured, then this video will give you everything you need to know. We will cover what pharmacovigilance means, why it is so important, what adverse drug reaction is, what is ADR or adverse event, how signals are generated, roles of healthcare professionals, pharmaceutical companies, and regulatory authorities, with real-time examples.
Let's start the journey of this learning from drug development. Drug discovery or development starts with target identification. Scientists identify biological targets like proteins, enzymes which link to a disease. Second is lead compound screening. Thousands of molecules are screened to find potential candidates that interact with the target. Third one is lead optimization. Promising compounds are chemically modified to improve efficacy, reduce toxicity, and enhance absorption. All the above-mentioned steps now handled with drug design methodologies.
Alright, once lead compound is identified, then the second step of drug discovery phase is preclinical testing. This is very critical phase as this involves laboratory and animal studies to evaluate safety and efficacy of the drug before even thinking about testing the same in humans.
There are two models used in preclinical testing. One is in vivo, another one is in vitro. What is in vivo? Whenever preclinical testing performed inside the living animal to stimulate human responses is called in vivo model. For an example, majority of eye-related medicines tested in rabbits, considering the anatomical similarities and physiological relevance. Second one is in vitro model. In vitro means experiments conducted outside a living organism. Typically this preclinical tests are performed in petri dishes, test tubes, or culture flasks using cells or tissues. For example, it is very difficult to induce or test cancer in animals and test anti-cancer activity of a drug in animal. In such instances, in vitro cell line models will be used for most accurate results. Alright, we discussed about the two models of preclinical studies. Let's see what are the key outcome of the preclinical studies and its significance. As we discussed, safety and efficacy are the two main components of the preclinical testing. For safety we will evaluate LD50, which is called as lethal dose 50. Dose in which 50% of animal population is died is called LD50. This mainly used to fix maximum dose for human population. To determine efficacy, effective dose or ED50 is identified during preclinical testing. Dose in which 50% of animal population produces the expected therapeutic response is called ED50 or effective dose 50. This helps to fix dosing and frequency schedule for human population. Apart from this, toxicities like genotoxicity, carcinogenicity, reproductive toxicities will be evaluated. Also, mechanism of action or pharmacodynamic profile and pharmacokinetic profiles will be evaluated in preclinical testing to understand absorption, distribution, metabolism and excretion profile of the drug.
After successful outcome from preclinical trials, company needs to proceed for IND application. It is investigational new drug application process. Let's see how it works.
An investigational new drug application is a formal request submitted to regulatory authorities to begin clinical trials in humans. The IND application is a critical regulatory milestone that allows a pharmaceutical company or researcher to begin human clinical trials. This application includes three main components. One is preclinical data, which includes all the previously discussed details. Second one is drug manufacturing information, which includes drug composition, formulation, stability and quality check. Third one is human clinical trial protocol, which includes study design, objectives, methodology, inclusion criteria, exclusion criteria, dosing plan and safety monitoring procedures.
Alright, let's assume that the company has submitted its IND application form along with preclinical study information. And it's reviewed and approved by regulatory authorities. Now it's time to start human clinical trials.
Before moving into human clinical trial, this is the right time to learn about the definition of pharmacovigilance. Pharmacovigilance, or PV, is the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problems. Just keep the definition in your mind for now. This will help you to understand the concepts in a very easy way. Alright, let's dive into human clinical trials. Human clinical trials divided into 4 phases. Let's look at each phases. Phase 1 of the human clinical trial focus mainly on safety and the dosage part. 20 to 100 healthy volunteers or patients will be used as participants to assess the safety, tolerability, and optimal dosage range of the drug. This phase determines if the drug is safer enough for further testing. Now let's recall some terminologies from the definition of pharmacovigilance. PV is related to detection, assessment of adverse events. Since detection of safety, tolerability starts from this phase, our road to pharmacovigilance also begins from this phase of the clinical trial. Phase 2 of the human clinical trial focus mainly on efficacy and side effects. 100 to 300 patients with the target condition will be used as participants to evaluate effectiveness and monitor short-term side effects. This phase determines if the drug is producing therapeutic effect in human population and also to monitor short-term side effects. Phase 3 of the human clinical trial focus mainly on both efficacy and safety in larger population. It's a kind of confirmation and comparison phase. 1,000 to 3,000 patients from multiple locations will be used as participants to evaluate efficacy, monitor adverse events and compare with existing treatments. This phase provides comprehensive data including efficacy and safety of the drug for further approvals. Before moving to Phase 4 studies, let's understand the concept and terminologies of pharmacovigilance. Let's recall the definition of pharmacovigilance again. It's deal with activities of detection, assessment and prevention of adverse drug reaction. What is adverse drug reaction? It's a harmful, unintended and noxious response to a drug when used at normal doses for a treatment or diagnosis. Now, in clinical trial, whenever this kind of problems happens to the patient, who is going to detect the adverse event? Yes, it's investigator. Post-detection, the information will be passed to safety department or pharmacovigilance department of the company. Assessment will be performed by PV team and based on the assessment prevention steps will be taken by health authority. Let's see how the assessment and prevention works in clinical trial. Let's dive into assessment part and see how PV team is assessing the report shared by investigator. Investigator shares patient details, investigator details, suspect product details and the adverse event details. Based on this data, PV team assess for the above 4 information for identifiable patient, reporter, product and event and make the case as valid individual case safety report in safety database. After making the case as valid case, pharmacovigilance team assess the seriousness, relatedness and listedness. Let's look at these 3 terminologies in detail. Seriousness is categorized as non-serious, medically significant, disability, congenital anomaly, hospitalization, life-threatening and fatal. Based on the impact it caused to the patient, investigator provides the respective seriousness in the report. Majority of the time, safety team goes with the seriousness provided by investigator. In special situations, safety team overrides the seriousness provided by investigator and the same will be reconciled between clinical and safety department. Relatedness is something like whether the adverse event happens because of drug or not. Let's take a complex example to understand this data element. A patient who is a chain smoker for last 20 years and diagnosed with non-small cell lung cancer. He is part of your company clinical trial which is discovered to treat non-small cell lung cancer. During the clinical study, after administration of your drug, the patient having severe cough and pneumonia. In this instance, how you identify whether the cough and pneumonia is related to smoking history or non-small cell lung cancer symptom or because of your drug. To identify this, clinical team and safety team use 2 critical methodologies to identify the relatedness. One is to challenge whether treatment will be stopped to check the adverse event. If the event is also stopped, then there is a positive relation which is called positive to challenge. Second one is re-challenge whether treatment will be reintroduced to check the reappearance of adverse event. If it's reappear, then it will be considered as positive re-challenge. In such situations, it will be decided that the issue is because of drug. Let's understand the listedness topic along with health authority assessment and prevention steps. Let's assume our drug is in phase 3 now. As per health authority expectation, apart from individual case safety report, company needs to submit periodic safety or aggregate report for every 6 months, starting from phase 1. During such instance, health authority will review and perform risk-benefit evaluation. Let's take our same example of non-small cell lung cancer and pneumonia adverse event. In this instance, though the adverse event pneumonia is serious, still it's okay considering the life-threatening disease condition of non-small cell lung cancer. So health authority will consider this event as an expected or listed event for your drug. During clinical trial, these events will be updated in IB label that is called as investigation brochure. Let's see one more example of a company drug which is in clinical trial for anti-inflammatory activity in patient experiencing cardiac arrest as adverse event. In such instance, health authority will assess the risk is more and the clinical trial will be stopped. Alright, let's proceed with our pneumonia example. Let's say you are handling a clinical trial case in safety department during phase 3. In this instance, you need to refer investigational brochure or IB label to document whether the event is listed or expected. If something unexpected or not listed in IB label, it will be documented as unlisted. Since we started about label, let's understand about some more labels to have better clarity. USPI United States Packaging Insert maintained by USFDA, which focuses the adverse events and its impacts mainly in United States population. SMPC, Summary of Product Characteristics maintained by EMA, which focuses the adverse events and its impacts mainly in European population. JPI, Japan Packaging Insert maintained by Japan, which focuses the adverse events and its impacts mainly in Japanese population. Though the label is different, the concept of listedness assessment is same between these labels. One more important terminology in pharmacovigilance related to clinical trial. It is SUSAR, SUSAR, Suspected Unexpected Serious Adverse Reaction. Hope we have all data elements to assess the SUSAR now. Since it's serious unexpected situation, health authorities will have an eye on individual SUSAR cases. It is almost like a breaking news to health authority for that particular product. Reporting perspective, pharmacovigilance ICSR have two modules of reporting. One is clinical module reporting, which is during developmental phase. Second one is post-marketing reporting, which is after drug reaches the market. Whatever we see as of now, which means till clinical trial phase 3, it is part of clinical trial module. After successful outcomes from 3 clinical trial phases, company needs to apply NDA form, which is new drug application form, to launch the drug into the market and to proceed for phase 4 study. NDA is a formal application submitted to regulatory authority to market a new product. While submitting this application, company needs to provide preclinical data, details of clinical trial on humans, drug manufacturing information along with quality control, labeling information and product samples. Once NDA is approved, the drug reaches the market. Now let's look at phase 4 studies and its significance. Phase 4 studies are also called as post-marketing surveillance. Since the activity handled after the drug reaches the market, it's called as post-marketing surveillance. During this phase also efficacy and safety is evaluated, but the only difference is this is very long-term and very large population. In clinical trial, some of the population like elderly patients, pregnancy patients, pediatric patients are excluded. However, these group of patients also will be included in phase 4 based on drug. Here we can basically divide the phase 4 into two. One is interventional clinical trial, which needs a kind of clinical trial setup. Second one is observational or non-interventional clinical trial, where no intervention required, as like clinical trial setup. Interventional category reports will follow clinical trial module in PV, and non-interventional or observational will follow post-marketing module in PV. These are the various type of examples covered under phase 4 setup. As we discussed, phase 4 clinical trial is designed as like clinical trial setup, but the only difference is it's going to evaluate the long-term efficacy or safety with comparison to other drugs. IPU is individual patient use, or CUP is compassionate use program, is a special situation. This is something like a clinical trial in individual patient. For example, let's take our example of the drug which is approved for non-small cell lung cancer. But in market, one of the physician have a patient with different cancer and tried almost all the drugs available in the market, and patient is not responding to any such medications. Physician understand the mechanism of action of your company drug, and based on that he want to try in his patient to save their life. In such situation, physician can enroll his patient into IPU or CPU studies to try the drug for the cancer which is not approved. This kind of studies called as individual patient use or compassionate use program. Apart from this, companies will conduct patient support programs to provide free education to the patient population, registry programs to patients or healthcare professionals. Apart from patient support programs and registry programs, market research programs also will be part of phase 4. Let's go back to the definition again. Pharmacovigilance is all about detection, assessment and prevention of adverse event. Now, in phase 4, who will detect and report? Yes, considering the different kind of setup, anybody can report. Clinical trial setup reports can be reported by investigator, and rest of non-interventional phase 4 reports can be reported by patient, doctor, pharmacist, nurse, any other healthcare professionals, vendors and anybody from market. And assessment will be handled by safety or pharmacovigilance department of the company. Prevention will be taken care by health authorities. Now let's deep dive and see how the assessment and prevention is happening in phase 4. Phase 4 assessment is little bit challenging as the report is received from both healthcare professional and non-healthcare professional. Let's see this in detail. As usual, the received report from market will be assessed for 4 mandatory elements. Identifiable patient, identifiable reporter, suspect drug and adverse event. If all 4 data elements are present, then the case will be considered as valid case. One important information to consider here is, in clinical trial scenario, we will follow the seriousness and important assessment factors based on investigator decisions. Since this report is from market, drug safety team will be accountable and responsible to handle assessment related to seriousness, relatedness and listedness. Seriousness, relatedness and listedness assessment will be made as we discussed in clinical trial scenarios. Once the assessment completed by drug safety team, individual case reports and aggregate reports will be submitted to health authorities. In clinical trial scenarios, aggregate report will be submitted every 6 months. In post-marketing, aggregate report will be submitted either 6 months or 1 year based on the number of years the product presence in the market. Health authority will evaluate the safety reports and perform benefit risk evaluation report. Post-benefit risk evaluation analysis, health authority will update all labels. Based on this, drug usage will be restricted to the populations based on health authority assessments. Apart from Phase 4, there are other reports also handled as part of post-marketing module. Reports received directly from patients and healthcare professionals. Each company's created different modes of communication like mobile application, toll-free number for calling, email, web reporting to collect safety information. Apart from this, patients and healthcare professionals reporting the adverse events directly to health authorities. These notifications will be forwarded to individual pharma companies and the reports will be handled as part of post-marketing module. Sometimes pharma companies will get report from lawyers. Also reports from literatures, conferences are also handled as part of post-marketing module. In terms of assessment, this will follow post-marketing assessment method.
Hope you learned about basics of pharmacovigilance. Alright, let's meet in next video with pharmacovigilance submission timelines.